Good morning, good afternoon, good evening. My name is Rigoberto Advincula. I'm a professor at University of Tennessee and a group leader at Oak Ridge National Laboratory. I congratulate the organizers of the Congress on digital nanobiotechnology and bioengineering. And today I'm going to talk about bio-inspired surfaces and templated patterning for tunable wearing and sensing. As the group leader of the macro group or polymer group at the Oak Ridge National Laboratory Center for Nanophase Material Sciences. We do a lot of work related to macromolecular science and engineering in collaboration with the Spallation Neutron Source, which is the best place to do neutron scattering experiments and investigating phenomena. On the other hand, I'm a professor at the Tickle College of Engineering, Chemical and Biomolecular Engineering Department. And at the University of Tennessee, we are known as the volunteers. So my research, a lot of that is primarily done at interfaces, surfaces, looking at nanostructuring of materials. Here is a representative of the work that our work that has been featured in the cover journals. And my group at the University of Tennessee is made up of undergraduate students, graduate students, postdocs, visiting scholars, as shown here. As editor-in-chief of MRS Communications, I welcome your best contribution as research letters, perspective articles, methods letters, or commentaries. Now, polymers at interfaces and their colloidal phenomena is akin to nanostructuring of materials. As polymers that can be tethered on surfaces where you can control the grafting density, the ordering, the hierarchy of structures, or even in micelles or aggregates or Pickering emulsions. Polymers are interesting because of the ability to control the chain conformation, constitution, configuration, as well as the different functional groups, whether it be optical, um, or even magnetic or living radicals, they have a lot of chemistry that can be rendered with soft matter. A lot of this actually involves nanostructuring or surface structuring of these macromolecules or even in combination with different types of physisorption and chemisorption, where we control the layer by layer deposition, the grafting of polymers on surfaces, or their electrodeposition. Another interesting phenomena is based on layering of materials based on electrostatic interaction, such as in the case of polyelectrolytes or polyswither ions that can be built up as layers or in the use of core shell, hollow shell particles. Another technique that is very important is the grafting of polymers on surfaces. This can be done by grafting from, grafting through, or grafting onto. Or in the case of grafting from, a lot of initiators are basically functionalized on surfaces and are forced to uh, pre- prepare them as chains that can be extended uh, through very high swelling regimes. Now, when it comes to films in nature, we are very familiar with the hierarchy of structuring and composition in the case of superhydrophobic films, which are naturally resistant to water based on the roughness or scale of roughness and the entrapped air, as in the case of the Casey-Baxter wetting phenomena. A lot of this can be observed as deviations from the ideal Young's equation, where you have a component of the roughness, which is the regime of the Wenzel equation, or the combination of roughness and trapped air, as in the case of the Casey-Baxter equation. One of the interesting things we did was to mimic the structure of the lotus leaf pattern in order to produce artificially superhydrophobic films that are bio-inspired. So in this case, we use molding or in particular PDM stamp where we grow polymers based on ATRP initiators. In this case, we grew a polymer that is based on polynipalm or polyanisopropyl acrylamide, which has an LCSD behavior around 37 degrees C. This means that this artificially mimicked lotus leaf pattern actually has a chemistry that is based on the swelling behavior or the super hydrophobic to a super wetting um, switching that is dependent on the temperature. So what I mean by this is, as you can see here, a surface that is flat, not patterned, will give you, of course, some LCSD behavior as shown here. When you heat the sample, you have a high wetting angle of 90, indicating hydrophobicity. On the other hand, cooling at 25 gives us a lower contact angle. But a surface that has a replicated lotus leaf pattern gives us a 153-degree angle or super hydrophobic effect, and about 0 degrees in terms of the wetting, indicating that of super wetting behavior. And this can be switched back and forth between super wetting to super hydrophobic merely by a 15-degree Celsius difference. So we have a mimic of a lotus leaf, but it's smarter than a lotus leaf. Another templating method we used was colloidal nanosphere lithography, where we would prepare colloidal spheres of latex particles using a Langmuir-Blodgett type of deposition and then growing conducting polymer on the shadows or the um, fringes of this array. So what you see here, for example, is a hexagonally packed structure of perfect spheres, and we happen to grow the polymer around it in a beehive pattern, half dome or full dome, or we can even harvest these capsules, as you can see here, after we prepare them by tensiostatic deposition. Now, these patterns are arrays of conducting polymers with holes in the middle. The holes in the middle basically exposes the substrate, and as you can see from the AFM and SEM pictures, we have a regular array of this conducting polymer. On the other hand, in the middle of this, which exposes the electrode surface, we can then grow an ATRP initiator, and then finally a polynipalm brush, which, as shown here, backfills or fills the array. And in fact, this polymer is even um, swellable, as I've shown earlier in the case of polynipam. And the conducting AFM here shows that the ridge pattern with the false green color has a heterojunction behavior. On the other hand, inside the pattern, which is the blue hole where we grafted the polynipam, they're basically insulating. Now, we have done since many chemistries on this. So, for example, you can reverse the patterning where the initiators are grafted on the beehive pattern. And then as you grow the polymer, as you can see here, there's an increase in roughness on top of the pattern, uh, also exposing the inner part of the beehive structures, again, amenable to grafting an initiator and then growing a polymer brush. So in this case, we have a ternary composition made up of the conducting polymer, the polymer brush that was first grown on the ridge pattern, and another polymer brush that was grown inside. Now, we serendipitously discovered that these are super hydrophobic films or materials when we overlay the deposition of another conducting polymer on top of this colloidal particles. By IR imaging, we can prove that we have both the polystyrene latex particle, which was the original material, and the polythiophene that was added on this material by potentiostatic and cyclic voltammetry methods. So the result is this roughness actually affects the wetting behavior. If we pattern them without the polystyrene, you can see the ester-containing polythiophene group having a decent hydrophobic effect. Once we add the 100 nanometer particle array, we have an increase in the contact angle. But then when we pattern it with the 500 nanometer latex particle, we observe routinely a superhydrophobic behavior. Now, what if you change the ester with an acid? You'll end up with a nice wetting behavior, but with patterning, it actually converts it into hydrophobic uh, surface as well, as can be shown here. So what's interesting is they hate water, but they absolutely love oil. So here, if we drop iodimethane, hexadecane, kerosene, cooking oil, they completely wet the surface with the oil. So these are what we call super hydrophobic yet super lipophilic type of surfaces. Moreover, the transition from reduced to oxidized behavior is accompanied by color change. This color change makes it an electrochromatic material or displays electrochromism. Moreover, the oxidized part is, as shown here, super hydrophobic, and the reduced part more wetting than ever. So the color orange or green allows us actually to visualize the wetting or non-wetting behavior of these films. Um, so let me turn to another technique that we use in our lab to do templating. 3D printing can be done by um, light projection, digital light projection, stereolithographic apparatus, or by extrusion based on fused deposition modeling or direct ink writing. So we have a lot of experience in this field. However, the first time I observed this type of features as shown here, with this resolution. So for example, you can see this naked woman on top of a hair strand of hair is patterning in the highest resolution. And this is usually achieved and was achieved back then by two photon polymerization. And this can be, of course, done today using this type of projection or stereolithography with two photon resonance as well. What I'm showing to you is that DLP, SLA are um, ways of developing features or pattern based on light interaction with the resin, which in this case resulting in cross-linking polymerization. So whether it's volumetric printing, continuous liquid interface production, stereolithographic apparatus, or digital light projection, they produce beautiful and clean parts as shown here, even without injection molding. So as a technique, this can actually go to even higher resolutions. So in this case, we did a project that involved the design of different surface features as shown here by SLA. So the question we were trying to answer was, can we produce super hydrophobic films on surfaces simply by 3D printing? And indeed, we looked at various types of features from spears to coil to square to a Mickey Mouse. And we found that at the right combination of factors, they produce a super hydrophobic effect based on the high contact angles here. Now, what this allowed us is to understand the nature of the printing, the tilt, the volume, the features or ladder effect, and the surface chemistry all contribute towards the fact that super hydrophobic films can be observed from 3D printed films or matrices, and that means we can control the wetting behavior. Now, let me turn to another type of templating, which is closer to the uh, logical phenomena of enzymes or different types of models based on rate model, occupation model, induced fit model to look at the interaction of substrate on protein dockets or surfaces. Interestingly, these proteins have similar size in dimension to dendrimers, which is a very interesting macromolecule with very regular and branch topology. So if we do this in enzymes and artificial enzymes, we use the principle of lock and key mechanism. So we have the lock and the key. let's say the key is available, which is the substrate or the template, but the lock is not available. So basically, what we have to do is construct a lock around the key. And this is involving chemistry where the original template is complex with monomers that can be cross-linked or polymerized, resulting in a cavity that's very specific for that original molecular template. We call this molecular imprinted polymers. And in this case, it mimics that of an enzyme substrate complex. The way the cavity is very specific for that particular substrate or original template. And so we've used this effect to design sensors. These films can be prepared on surfaces that include a surface plasma resonance spectrometer, an electrode surface, or even a electrochemical impedance uh, spectroscopy working electrode. So in this case, you prepare a substrate or your analyte, surround it with monomers that we actually studied in terms of their particular electropolymerizability. Electropolymerize the complex, which leaves you this templated structure. And by removal of the substrate or chemical, you're left with a cavity that is very specific to the original template. So using this principle of MIP or electropolymerized MIP, we were able to design sensors that have high sensitivity and selectivity. So in this first example, you can see how we try to template different drugs whether it's theophylline or paracetamol or naproxen. We use polythiophene as a matrix or a monomer to surround the original template, electropolymerize it on the top of the gold surface for a surface plasmon resonance detector scheme. And then, as you can see here, this system has a very high selectivity to the original template, which was used for making the sensor, whereas the rest were left behind. So this, you can see how powerful it is to use the lock and key mechanism to design artificial sensors based on electro polymerizable molecular imprinted polymers. And we have been busy trying to demonstrate it on various types of analytes, ranging from organophosphate or serine analogs or dopamine, uh, bisphenol A, melamine, morphine, aspartame, and we've seen this demonstrated even on oligopeptides associated with the dengue virus. As you can see here, we can combine this with the colloidal nanosphere lithography and molecular imprinted approach to produce high um, surface to volume surfaces that are capable of sensing or being templated to make sensing. So in this case, we have again this monomer templating here a norepinephrine enantiomer. And these two, with these two, only one is actually active as a drug. But we can template for each one and then use this templating as a way of sensing the active and non-active complex. And this time we actually use quartz crystal microbalance. So they do work. The sensitivity and selectivity was shown by the response on the frequency. So for example, depending on the analyte, we can switch back and forth between the two enantiomers in terms of selectivity, or if we try to compare it with templated or non-templated, we clearly see that the templated part gives us the high sensitivity. And then when we compare it with other drugs like naproxen and dopamine, again, the original template gave us the highest sensitivity. And then finally, we demonstrated the efficacy of solvent washing to produce more cavities. So let me switch to the final part of the talk, which is focusing on viral delivery agents or viral sensors. Virus here means the harmless, relatively harmless cowpea mosaic virus and other plant viruses, in this case, the icosahedral CPMB or cowpea mosaic virus. We then functionalize these viruses with different groups, in this case, a reactive group capable of polymerization as a polymer brush or capable of hosting with this branch dendron the DOTA or other complexes with is capable of trapping platinum and then releasing this. So in this case, we actually loaded the CPMV virus that was modified with platinum particles. This is to demonstrate, in this case, we can have a drug or a particle or another dye which is capable of treating or killing cells or even cancerous cells. So in this case, we were trying to demonstrate its ability against macrophages. So we functionalized the virus with groups and then complex a drug. In this case, with photodynamic therapy, we actually have been able to show that the ability to load these surfaces with drugs and apply a photodynamic therapy procedure was very effective. So the virus was modified by adding a linker and then growing the polymer dendron on the surface or doing click chemistry to attach the dendron rapidly. The result is you get a phthalocyanine dye that you can load on the cowpea mosaic virus and then use this dye loaded structure or phthalocyanine dye loaded structure for photodynamic therapy, which means they're active with light and then they can produce radical oxygen species or simply be able to destroy up to the cellular cell membrane level the cancerous cells. These are some of the tests. These are some of the uh, TEM images showing the very high resolution we can get to distinguish the virus from the matrix. The absorption here simply shows that these viruses also have the loaded model drug. And then finally, we use live cell dead assay, live cell dead cell assay in order to differentiate its ability to kill cancer cells. And as you can see here, we're able to show very high um, death on the uh, cancer cells, melanoma cell line, and shown by the dead cell count, we can control between uh, the virus that contains the branch polymer or carrier of the uh, psilocybin material or without the virus or simply dead control. And here we can clearly see that we have a lot of efficacy in terms of reducing the cell viability with the higher concentration of the loaded drug. Finally, I can demonstrate that this cowpea mosaic virus nanoparticles are capable of forming films. So in this case, if we go back to the full templating with colloidal particles and then grow uh, the uh, polypyrrole around it, remove the original template, and then finally introduce the viruses. The question is will they go and will they end up forming layers? And the answer is yes. You can see the viruses here basically surrounding the holes of the arrays, indicating that the viruses would like to settle on the surface as well. And we can even liberate these patterns as films or freestanding films, showing that it's possible to have a very high concentration of the drugs in a templating manner or procedure. So with that, I'd like to thank you. Thank you for the opportunity to give this talk and dear's time. I'll be happy to answer questions. If not, feel free to email me with my email at the University of Tennessee and be happy to correspond with you. Thank you.